In the last objective, we talked in general about the receptor in terms of the anatomy of the receptor, how it's a specific, highly specific to a certain ligand. And we also talked about regulation of the receptor as it relates to its surrounding environment. In this objective, we focus on the different types of receptors. This is important because there are different types of ligands. So, which ligands bind to the different types of receptors? So, here we're talking about four main types of receptors. So, these include the ion channel linked receptors, the G protein linked receptors, enzyme linked receptors, and intracellular receptors. So to start, the ion channel linked receptors are also known as ligand gated channels. These are found in the plasma membrane. So that's shown here, this large protein present here. These are going to convert chemical signals into electrical signals. So as shown here, we have a particular signaling molecule. In this case, most of them can be neurotransmitters. So in the example given here, the signaling molecule represents acetylcholine. Acetylcholine is a neurotransmitter. And once it binds to the receptor that's embedded in the plasma membrane, it's going to trigger opening of this ligand gated channel. And this will allow positive ions to flow through the channel. And these positively charged ions will trigger or will allow the cell to produce electrical signals. Okay, so to recap, these ion channels open or close in response to binding of a ligand. In this case, you have the red that represents the acetylcholine binding to the receptor and allowing the positively charged sodium ions to flow through into the membrane, and that is shown here. Next, we have the G protein linked receptors. So G protein linked receptors, this is shown in blue. These are transmembrane proteins with a binding site for a signaling molecule on the outside of the cell. And it also has a binding site for a G protein, all right, that extends into the cytosol. And so what this does is the G protein coupled receptor, it couples the signaling molecule to signal transduction pathways on the inside of the cell. Now, the signaling molecules range for the G protein linked receptors. These are super diverse and they bind a diverse number of signaling molecules. So G protein coupled receptors are important in vision, sense of smell, regulating mood, behavior, and immune system. So they're going to bind molecules that are specific for those areas. For example, vision, you have rhodopsin receptors that are considered classified as G protein coupled receptors. These are sensitive to light. In terms of the sense of smell, chemicals in perfume can bind to G protein coupled receptors and trigger a response. And so this will allow you to smell if something is good or something has gone bad. So G protein coupled receptors, very diverse. There are more than 400 are potential targets for pharmaceutical interventions. So in terms of the overview of the mechanism, when the signaling molecule binds to the G protein coupled receptor, it triggers a conformational change and allows for a G protein to bind. And then that G protein will also be allowed to bind to another nucleotide that is responsible for other biological processes in the cell, okay? And so we'll get into that a little bit later in the next objective. Our third type of enzyme linked receptors are enzyme-linked receptors. So these are transmembrane receptors that can bind signaling molecules on the outside of the cell, and it has an enzyme component on the inside. So extracellular domain and an intracellular enzyme domain of the receptor is shown here. So these are responsible for binding hormones such as your insulin and your growth factors. Examples of enzyme-linked receptors include your tyrosine kinases. And so in terms of the mechanism of action for these tyrosine kinases, all right, um, ligands will bind and cause these receptors to dimerize, okay? It's gonna cause these receptors to dimerize. In addition to that, these kinases will catalyze the transfer of a phosphate group from ATP to the tyrosine part of the protein, okay? So you have two important events that happen. So ligand binds cause dimerization, and also you have the transfer of a phosphate group to the tyrosines. So once these kinases are phosphorylated, this will cause the kinase to be considered active. All right, this will cause the activation of the tyrosine kinases. And then finally, you have receptors that are located inside of the cell. So your intracellular receptors, these are found in the cytosol or the nucleus. And these are mainly going to be transcription factors. And so an example of an intracellular receptor would be the estrogen receptor. And these are going to bind ligands such as estrogens. All right, estrogens are have been linked to breast cancer. So estrogens binding to estrogen receptors and different estrogen receptors have been linked to breast cancers. Signaling molecules have to be hydrophobic. So this should be a review from the previous learning objective. In order for the ligands to bind to the intracellular receptors, they have to be hydrophobic. So some are going to combine with receptors in the cytosol, and then these will these bound receptors will move to the nucleus. And eventually they're going to bind to the DNA inside of the nucleus and trigger some biological events such as gene activity in gene activity. So overall for this objective, we've learned about the different types of receptors. The different types of receptors include your ion channel linked receptors. These are going to respond to ligands, such as neurotransmitters. These can bind these channels and allow for ions to flow through. We have G protein linked receptors. These are very diverse receptors. So these are going to be receptors that contribute to vision, sense of smell, changes in mood or behavior. So they're likewise going to bind to ligands that are responsible or that are going to activate those behaviors. Or those changes in mood or behavior, changes in vision, changes in other things, all right? And then our enzyme linked receptors, we gave an example of the tyrosine kinase receptors. Ligand will bind to the receptors and cause dimerization of the receptors, so causing those receptors to come together. And then this will also, with the enzyme linked receptors, they will have phosphorylated um, or enzyme linked domains in the intracellular portion of the receptor. And so those will be phosphorylated. So a phosphate group will move from ATP to those to that particular intracellular domain. And then you have intracellular receptors. Your intracellular receptors are located in the cytosol, And these will bind typically, we gave an example that these will bind to hydrophobic molecules, okay? These are going to bind to hydrophobic ligands. And the example that we gave is the estrogen receptor is a intracellular receptor and it will bind to estrogens. These receptors will then move into the nucleus, bind to the DNA, and alter the gene activity in some way.